Thanks, everyone, for attending the Society for Immune Polymorphism's 2025 Winter Webinar. For those of you who are not familiar with the Society, we are an international membership organization dedicated to understanding the genetic and functional variation of the vertebrate immune system and the immune system's role in evolutionary biology, disease, and health. If you're not yet a member, you can join the Society by going to tinyurl.com/jointhesip, and I have put that link in the chat. Today's webinar is titled HLA Region Assembly and Genomics. If you have questions for the speakers, please enter them in the Zoom chat. Finally, I'm pleased to introduce Professor John Trowsdale, who will be moderating the webinar. Dr. Trowsdale graduated at the University of Birmingham, where he also studied for his PhD in bacterial genetics. After postdocs in Paris, La Jolla, and Oxford, he joined Walter Bodmer's lab at the Cancer Research Fund in London. In 1997, he moved to Cambridge, UK, to be a professor of immunology. He is now emeritus and spends his time writing. His first book, Guide to the Immune System, is called What the Body Knows and is published by Yale University Press. Thanks very much, John.
First speaker is Stephan Scholz, who graduated with a master's degree in physics from Humboldt University Berlin in 2014. Since 2017, he has been involved with the assembly and characterization of HLA regions in Dr. Alexander Dilthey's lab at the University of Hospital Düsseldorf in Germany. That's work in close collaboration with the lab of Dr. Paul Norman at the University of Colorado. So, Stephan, the floor is yours.
Okay, I'm happy to talk about how we compose a reference MHC sequence for scaffolding. So together with Paul Norman, we do de novo assembly of MHC sequences. And the assembly program typically outputs not an end-to-end sequence, but several contigs, the order of which is unknown. And to order these, we use a reference that we map against, and also this reference can be used to fill in gaps at the border. Okay, let's jump right in. So what we have is a set of haplotypes from the population and some reads that we want to construct a new haplotype from. And what we want, is the new sequence. So how might we go about this? So let's say we capture the structure variation of the population in a genome graph. That's here. And then we take the reads and map it against this graph, and then just pick the variations where the graph land on. This approach is simple, but it has a few shortcomings. The MHC is known to have some duplicated genes, and the reads may map to variants where they did not originate from because the genes are so similar. Examples would be the C4 genes or the DRB genes that are very similar. And this could be resolved by using statistical information from the population. But this statistical information, how the variants correlate, is lost in this graph. We can regain this information by putting the haplotypes back into the graph. You would then call this a colored graph. And there are more complicated graphs that capture higher order relationships like translocations or inversions. But in the case of the simple graph that only contains bubbles, this would be equivalent to a multiple sequence alignment. So you can think of it as taking the graph and stretching out these bubbles and inserting gaps in the multiple sequence alignment. So down here, it's a representation of a multiple sequence alignment where the sequences align, they are on top of each other, and the white regions are the gaps. This brings us to our first problem. How do we generate such a large multiple sequence alignment? Because we will use approximately 130 haplotypes from the Human Genome Structural Variation Consortium. And each of the sequences is approximately 5 megabases long. This alignment would take more than a week. We actually never let it finish. And yeah, the computational complexity is polynomial in the length of the sequences. The exponent is smaller than 2, but larger than 1. And this nonlinearity gives us a chance to cut down the runtime by splitting the sequences into shorter pieces and aligning the shorter pieces. So since we annotate these haplotypes anyway, we use genes to cut down the sequences, and we select genes that are stable in its order, and then align the genes separately and the intergenic regions. This also has the advantage that we can split this alignment across multiple machines, so they can run in parallel. After these regions are aligned, we can then concatenate the alignments. So that solves the problems that generating large MSAs takes a lot of time. So then we have to find a way to pick out the structure variations. Yeah, I want to point out that picking different structure variations is akin to switching the color in this graph. And in this multiple sequence alignment, we cannot switch between the colors at any position. You can see my cursor, right? Okay, I will assume you can see this.
Yeah, we can see it. Yeah.
Okay. For example, you cannot switch at this position because that would mean that in this graph you take this variation and then go back here and then take this variation, which is not something that we observe in the population. So we do not want to make this a possibility. But it's always possible to switch the color in these aligned regions. Okay, on another thought, we had this. I pointed to the problem that reads will make the regions where they did not necessarily originate. And here we take the example of the C4 gene. It's approximately 20 kb long and can occur between 1 and 4 times. And if you don't have spanning reads, then it's hard to determine the exact number. So this is depicted here. This is supposed to be a multiple sequence alignment. The green, the yellow boxes are the C4 genes, and I left out the gaps to make it more easy to see what's going on. And next to it, I marked some SNPs. So the rows are the haplotypes. Above it are the SNP alleles, and below it here, these are the alleles of the read. And we can now use as a correlation between the SNPs and the number of C4 genes to determine the number. Yep, so in this case, the most likely candidate would be haplotype number 3 because we only have 1 deviation in the alleles compared to the reads. And we will not only use this technique for duplicated genes, but for every structure variation. So an advantage of this is that the SNPs that we use are easy to find in a multiple sequence alignment, and we only will select the cleanest SNPs that have clean alignment on both sides. So with this, we get the following model. Again, this is a multiple sequence alignment with gaps, and the dotted lines are SNPs that we found in this multiple sequence alignment. And we can now think about starting on the left side and walking along this multiple sequence alignment and start copying from one of the haplotypes. And at each SNP position, we compare the allele of this haplotype with the reads and check whether they agree or if it would be better to switch to a different haplotype and start copying from this different haplotype. And in this way, we can get our scaffolding reference. This model lends itself to be modeled as a hidden Markov model. This was done in 2003 by Lee and Stevens. And it has the following properties because it copies always segments. This is close to nature, as nature, recombination in nature also happens segment-wise. As I said, the structure variance is inferred from framing SNPs, and this model will take into account the recombination rate. That's a constant that you can, this characteristic for the population that you can calculate. The read support, so regions where you have a lot of reads will be weighted higher, and the read error rate that is dependent on the technologies that you use. So nanopore would have higher read errors than Illumina, for example. Here is a mathematical formulation. I do not expect you to fully grasp this in this short amount of time, but yeah, I will point out a few things. So as a reminder, in a hidden Markov model, you have several states that cannot be directly observed. In our case, these are the haplotypes that we copy from. But what you can observe is some observable. In our case, it's the alleles of the reads. And typically, each of the states can produce the observed value, but with a different probability. So this would be the emission probability. And what this basically says here is that you take the number of reads that do not agree with the haplotype at this position and multiply this. So this would be the number n and multiply it with the read error rate. And you do the same for the reads that agree at this position. So you get a probability that the haplotype emitted the reads that you observe. And what the Lee and Stevens model did elegantly is a transition probability. So that means the probability that you stay in the haplotype or switch to a different haplotype. And this is shown here. What is I want to point out a few things that make sense. So first, the probability that you do not switch to a different haplotype, so that you stay in the haplotype that you copy from, is always greater than the probability that you will switch. The probability that you stay in your haplotype is higher the more haplotypes are in your set. This also makes sense because the more haplotypes you have in a starting set, the more likely it is that the new sequence will have a relative in the set that you start from. The other thing is this factor 1 minus e. E is defined here, contains the recombination rate that is constant, and this delta term is a base pair difference between two SNPs that you have to between the two SNPs in question. And this 1 minus e term has a shape that is depicted here. So if the SNPs are next to each other, it is unlikely that you switch. But if you consider a larger distance, the more likely it becomes that you switch to a different haplotype. Okay, so with these probabilities, you can, for each pass, calculate the probability that this pass emitted the reads that you observe. And we have to maximize the probability, and this will be as a pass that we will take as a reference. And there are a lot of passes, and how to select the best pass is a computer science famous algorithm, the Viterbi algorithm. Here's an example computation, but I will not go through it because I think it will take too much time, but it's in the recording, and if you could go through it yourself if you so wish. But the essence is early on you discard passes that you can know that will never become the most probable. Okay. So now that we have the model, how do we use this in to obtain the scaffolding sequence? So first we do the multiple sequence alignment. And in the multiple sequence alignment, we search for all the SNPs and we remember them. And then we take all the 130 haplotypes and add them to the complete human genome. So this is depicted here, only the MHC haplotypes. And in each of the haplotypes, you can see here marked the SNP positions that we obtained from the multiple sequence alignment. Then we take our reads and map them against this extended HD38 reference. And by this step, we also filter out reads that do not map to the MHC, but to other parts of the human reference. And they will scatter across different haplotypes. That's okay. For each read, we can then read off the SNP alleles and put it into our HMM calculation. And out we get the scaffolding reference. So to evaluate this, we did, here's an intermediate result. We took nanopore reads and removed one of the references and tried to recover the sequence with our HMM approach. The nanopore had a depth of approximately 10 to 20 and were not called in super high accuracy mode, but it just the normal calling. And you can see here the base pair difference that of the sequence that was obtained via this HMM compared to the sequence that we took out. So which we think we know the real sequence because we put in a lot of effort to assemble it with several technologies. And as a comparison, there's a base pair difference to a state-of-the-art de novo assembly. And you can see that it performs quite favorably. So it's always lower. Sometimes it's in the same order of magnitude. Still, the distance is quite high and not good enough to build MHC references. And here, Nicholas Pollock came up with the idea to use the output of several de novo assemblers and take the contigs and map them then to this or to any scaffolding sequence. And what he found was that the weaknesses of the different de novo assemblers basically cancel out, and you then get near perfect assemblies. So this is probably the approach we will take in our project. Okay. We also extended our model to allow for mutations. So one of the problems that can occur is that the reference, the scaffolding reference, may have a real mutation in one of these SNPs. And if you have then a lot of reads at these positions, these reads are each its own factor, and they will clamp hard down on this on a non-matching haplotype, so that you will get unnecessary switches from one haplotype to another and then back again. To reduce this, we introduced sub-states. So each haplotype now has the possibility to have mutation bases, but the probability to go into one of these substates is very low. And in the next step, the probability to go back to the haplotype is pretty high. So this models mutation, single base mutations in these SNPs. Another extension is that so far I only talked about the haploid case, but this model can also be extended for the diploid case. In the diploid case, you square the spade state. So a new state now would be that haplotype 1 is in in state 1 maybe, and haplotype 2 would be in state 3 or something. So that would be one state. And the runtime of this model grows then quadratically. But this can be done, and you get out some two scaffolding sequences. And yeah, we did this. And this, what I just explained, is a finished program, and it's about written in Rust. Rust is known to be in the same efficiency class as C and C++, so it should be quite performant. And as an input, it takes a multiple sequence alignment as a FASTA file and some file where you map all the reads to this extended HC38C reference. Okay, yeah, with this I can already come to my summary. We employ the SMP-based Lee and Stevens model to create scaffolding references for de novo assembly. The references are a mosaic of already assembled MHC sequences. And in our model, we take into account the known variations of the population, the recombination rate of the population, read support, so that if you have more reads in some regions, they will be weighted higher, the read error probability that is technology dependent, and the mutation probability. And I would say this model is especially useful if you have sparse or error-rich data, because in that case, it is advantageous to take into account prior information from the population. Yeah, I want to thank Alexander Dalte, Paul Norman, and Nicholas Pollack for doing this project with me, and Lisbeth Gütlein and Kristen Bade for being in our weekly talks. And I'm open to question now.
Thanks very much, Stephan. I'm just trying to see if there's any questions. There's no open questions in the box. Could I ask you, though, what is the error rate currently in nanopore sequencing?
Yeah, that depends on how much effort you put into base calling. Honestly, I'm a bit nervous and I forgot about this. I think it is 0.2% or something like that.
Mm-hmm. Much improved then.
Yes.
Okay. So if there are no further questions, let me see. So if there are no further questions, let me see. There's no further questions in the box. I think we'll go on, continue on to Kristen's talk. Okay, thank you very much, Stephan.
Pleasure.
So Kristen Wade obtained her Bachelor of Science degree in Bioinformatics at Virginia Commonwealth University in 2014. She completed her doctoral thesis work in the laboratory of David Pollock at the University of Colorado in 2022. Her thesis work focused on understanding and modeling the evolution of the non-coding genome. In 2022, she began her postdoctoral work with Dr. Jill Hollenbach at the University of California, San Francisco, investigating the genetic architecture of multiple sclerosis across the MHC. So, welcome, Kristen. We look forward to your talk.
So I'm going to be sharing with you all today some of our recent results investigating genetic risk for multiple sclerosis across the MHC. And this is sort of phase 2 of our recent analysis on this project. And so I'll get into how it's looking and some of our current results. Okay, so multiple sclerosis is a neurodegenerative disorder that's believed to be autoimmune in nature. And we've known about it for quite some time. Some of the initial studies, genetic studies on it, date back to the 1970s, where we discovered significant roles for the MHC class 2 region. Specifically, HLA-DRB1 1501 has been the dominant MS risk allele and the primary genetic component that we've known about in terms of conferring MS risk. Of course, many diseases are complex, and so there are also roles for environment, and then the biology of the immune response. And so MS, is basically the intersection of all of these very complicated interactions. And though it seemed like the genetic component was simple and we had it squared away with DRB1 1501, turns out in a recent study in 2019 from the International MS Consortium that there are actually 200 genome-wide non-MHC risk loci for MS. And even within the MHC, they identified an additional 32 independent signals beyond just 1501. And so this really expanded kind of our perception of the genetic risk structure of MS and has really opened up the need for much more sensitive and focused genetic analysis to really elucidate what's happening in terms of genetic risk. And so this is from that same study, focusing in now just on the MHC and what they found there. And so we have our orientation. Here's the chromosome, and here's the MHC. From top to bottom here, it's sort of vertical. Usually we think of it as horizontal. And so, of course, we see the biggest risk signal with 1501. But even when they accounted for 1501, they identified all these 31 other signals, many of which were also non-HLA and intergenic variation. And so this, again, underscores that there is quite a bit more to be extracted from the MHC in terms of MS risk. I'll also add, too, that this study was carried out as a meta-analysis of several, it was about combination of, I think, 3 SNP genotyping chips. And it was performed exclusively in European American ancestry populations. And there's, due to the differences in chips and sort of inconsistencies between those, also involved imputation of variation as well. And so ideally, we can try to get beyond the level of genotyping chip and actually get single nucleotide resolution across the entire MHC, which has kind of been the goal of our work. So we'll get into that in a bit. And then just at a population level, as I mentioned, that study was exclusively in European American populations. However, we know that African American populations also get MS, and it presents slightly differently in those populations as well. As you can see here, there's quite a high prevalence of it in African American or Black communities. Even though for a long time it was considered a purely European disease, we're now realizing that that's not the case. We also know that your percent of European versus African American ancestry can also impact how risk plays out as well. And so, though we know these things, still there has been very little work done to actually solidify how we can interpret that in the context of MS. So with all these things considered, our goal with this study has been to take targeted high coverage sequencing in our transethnic cohort and try to discover both novel variation and then further resolve known MS risk signals within the MHC. So at UCSF, we have this really excellent department for studying multiple sclerosis, and this group was established in 1989 and have been both treating and collecting data from MS patients since then. So these are all individuals from the Bay Area, California, that have come here seeking treatment, but we'll be describing them as European American or African American backgrounds. All of these cases have been diagnosed by neurologists, and the vast majority of these are relapsing remitting cases, which is one of the subtypes of MS. And we have in this cohort age and sex matched controls with no history of chronic disease. And the DNA we're using in this case is collected from peripheral blood mononuclear cells. So our sample size is about 1,600 individuals, and you can see the breakdown here of how that plays out by population and case control status. So we're looking at about roughly 750, 850 individuals per population for our association study, which again is nowhere near the 30, 40,000 individuals they had in IMSGC study, but because we have such high quality sequencing data for these and such high quality disease information, we're hoping to be able to expand our understanding of this region. And as I was mentioning, this is sort of a display of the kind of sequencing depth that we're able to access using a target capture short read approach. It was originally developed in the Parham lab by Paul Norman, and then Danilo Augusto has also expanded and enhanced this approach to be even more sensitive. As you can see, looking across the MHC here, we get really strong enrichment across the whole region with this target capture short read approach. So, as we just heard about from Stephan, it is not a trivial task assembling and interpreting variation, even less so with short read data. And so to that end, sort of the first half of my work with Jill was developing a de novo short read assembly method called MH Constructor to assemble short read data in a reference-free manner. And so using that, we're able to generate these assemblies with our data. Following assembly generation, I performed haplotype-informed scaffolding, error correcting, and then assignment of heterozygous sites. These were then aligned to correspond to the HG38 reference. Again, this is not an alignment-based approach, but once the de novo assemblies have been formed in a reference-free manner, we then have to convert it back to a single scale so that we can compare consistently across populations. And so I used the HG38 reference MHC for that purpose. To add kind of an additional level of trying to get as much information out as we can, I also use Pangini, which is a graph-based variant caller to incorporate with our de novo assembly output as well. Finally, variation was evaluated for possible false positives because we know with short read data that can be a big problem. And so I've done this by taking the 90 phased MHC haplotype graph structure from the Human Pangenome Reference Consortium, and essentially, if our variants were included in that graph, they were retained, and if they were not included, they were excluded. So we do lose a few true positives doing that, but we filter out significantly more false positives. So it's definitely a more conservative approach that can help us be more confident in our ultimate variants. And then, as we know, short read data especially struggles with the HLA genes. Because of this, we didn't want to reinvent the wheel on those parts of the genome, or those parts of the MHC. So we used the NGS engine software to perform second field high resolution genotyping for the HLA alleles specifically. So now to kind of validate, show some validation of this approach, we use the Genome in a Bottle MHC benchmark to compare to our predictions. And so I generated simulated data for 60x short read from the two-phase haplotypes for that Genome in a Bottle individual, and then ran it through the analysis and compared to the benchmark variant annotations. And this is also sort of a breakdown of how both these methods of variant calling perform, Pangini and then our MH Constructor de novo assembly. So they collectively identify 13,000 SNPs that are the same between both methods, which is nice to see. And then interestingly, they both identify SNPs that the other one does not. So we find about 2,000 in Pangini that aren't in MH Constructor and about 3,000 in MH Constructor that aren't in Pangini. So combined, we can access roughly 18,000 of the 22,000 true SNPs in that MHC benchmark, which significantly expands our access to variation. And then I'll also add we can end up with a relatively low false positive rate. And I'll give you, how that false positive, true positive rate plays out across MHC, right? Because we know certain regions are more problematic than others. So at the top, we have percent recovery of true SNPs from the benchmark. And as you can see, the vast majority of these regions are well above the 99% recovery threshold, which is great. We do see a couple that fall slightly lower than that, likely in the HLA genes themselves. And then, of course, we do see a drop specifically around the class 2 region. Even with both methods combined, short read just still struggles to identify that. And that specifically falls between the DQA1, DQB region. And then in terms of false positives, we see a reassuringly low presence of those, again, through almost the entire region, with the exception literally just between DRB5 and DRB1 is where the vast majority of them occur. So just knowing that as we interpret our data to kind of be hesitant about interpretation of that region without more follow-up and validation. But overall, really confident results in our ability to identify true variation here. So using all these approaches, in the end, we actually are able to about double the number of SNPs that are accessible to us for MS association testing as compared to the previously used genotyping chips in the IMSGC studies. So there was the 2019 one, and then there was a more recent one in 2023 as well. This obtained up to about 35,000 SNPs. And then in these two columns here, we have our data, which reaches about 65,000 SNPs that are accessible. Also adding again that we have now also the African-American population, which we can do this in, which was not performed in the previous study. So our statistical model for evaluating the MS signal here, of course, we have our covariates. We need to account for 1501, and we need to account for sex because we know that MS is sort of sexually dimorphic and is at your much higher risk if you're female. So we account for that. And then we use only the independent sites of the MHC to account for population structure. And then we also generate a kinship matrix to represent genetic relatedness, which again is based off of just the independent sites in the region. And then due to the sort of known differential patterns of allelic diversity across at the HLA and the MHC, we do consider we'll be testing the European American and African American populations separately. So after all this, this is our Manhattan plot across the MHC. You can see the African American here on top with your European American below. And so, you know, this looks like so many dots. You can see some things that look like peaks for sure. And we see, again, for our sample size that we have access to reasonable enrichment of signal. But part of the issue that I've spent quite a bit of time on has been trying to figure out, okay, how do we go from this to actual target or candidate SNPs or regions, very precise regions of the genome to focus on. So one of the ways that we thought about this, so here's kind of the volcano plot version of that, where you have the significance here, which was on the horizontal, it was on the y-axis in the previous slide, but now instead of genomic position, we're showing odds ratio. So this kind of gives you the spectrum of risk and protective effect of the SNPs that are coming out with relationship to their significance. So things that are more like outliers are going to be the ones that are generally going to be our signal. And so this is for the whole population, each separately. But another way that we try to think about this is because we know DRB1 has such a strong signal, because we know that there is so much LD with it across the region, we did, in addition to separating it as a covariate, which you saw before, we also ran an analysis where we separated individuals that had a DRB1 allele from those that did not. And so we can see here in both populations for the group of individuals without the 1501 allele and the same signal that I was showing you. And then here we have the European American individuals with 1501 still. And you can see the population or the case control values, of course, are smaller now because we have subset the population. Interestingly, this has a pretty strong effect to boost our signal detection ability, especially in the individuals lacking 1501, which is really encouraging that it's indicating that this is a good way to actually tease out 1501 independent signal even further, and sort of reaffirms our choice to stratify these in this way. And so now looking at just the populations of individuals lacking 1501, if we sort of focus in on these and try to look at some of the signal and the regions, the genomic regions where that's coming from, we can identify sets of shared variation or shared MS signal between the African American and the European population. And so this over here, I've just listed the genomic region that corresponds to the lead SNPs that have been identified here in the same color. And so by comparing, so because we know that African American populations tend to have lower LD than European American populations, by comparing the two and looking for ones that occur in both, we can be even more confident that what we're seeing is actually independent signal. And I'll also just point out as well, these regions with asterisks are also the regions that are identified as one of the 32 independent loci from the 2019 study. So it is reassuring that we are also picking those up with this method too. And then as we were, wondering about with this approach, can we pick up more variation that is not on chips? And it turns out that each of these sort of top or lead SNPs identified here are unique and not found on the genotyping chips from the previous IMSGC 2019 study, which again is just an indication that we are able to access much more fine-grained resolution of what's happening in this region than just using genotyping chips alone. And so, of course, we've identified these regions. Now we want to kind of ask what might they be doing. And it turns out that a lot of these have strong potential for being regulatory elements in terms of gene expression. So in this first column, we evaluate whether they fall within a promoter, an enhancer, or an acetylation peak. In the second column, there's a previous study that came out that looked at differential methylation in MS across the MHC and identified sites that were differentially methylated. And so we also looked at whether they fell near to those sites too and could potentially be involved in that differential methylation process. And then these last three columns represent EQTL data from GTEx in three different cell types: in whole blood, in nerve, representing the CNS, and in the brain. And so, as you can see, again, there is very strong potential for cis and potentially some long-range gene expression impacts as well. So to kind of dive even deeper into one of these regions specifically, so this is kind of what we find. And this particular one was found both in the two groups lacking 1501, but it was also found as a peak in the European population of containing 1501 individuals as well. So this would be an example of a signal that is likely consistent for all of MS potentially. All MS architecture might involve this particular region. And so this vertical line here indicates the African American SNP that was identified with this signal, and as you can see, it falls directly into a candidate or a cis regulatory element, and also overlaps with acetylation peaks as well. And as you can see, it's very closely upstream of DQA1. And so we know it has EQTL activity as described in the previous slide. But something else that we also looked at that we found several examples of in these results was that many of these SNPs are likely to have splicing activity or splicing control over much more distal genes as well. As you can see here, an example from this SNP. We have the class 2 genes the class 2 genes here, and in whole blood and in CNS, all of these have strong splicing activity across this region, which suggests possibly another level at which class 2 genes could be impacting risk of MS, not just through like the function of the protein, but also what's happening in terms of alternative splicing composition during the expression process. And then, so now we were just talking about signals that are found in both African American and European American populations. So now we look at some examples of ones that are only found in the European American group. As you can see, some of these are actually some of the strongest signals in the European American group. And we find 3 examples of this as well. And these also were not found on the genotyping chips. To just look at one of these in kind of a little bit more detail, and this is a somewhat novel locus. It was not included, I think it was outside the range of the 2019 study. This between scan 3 and trim 27 also exhibits strong EQTL potential. You can see here the sort of CNS tissues control this ZNF603P gene, whereas in whole blood, it seems to be controlling ZFP57. And so ZNFP603P has been linked to brain aging and schizophrenia in previous studies, whereas ZFP57 has actually been shown to exert a sexually dimorphic control over genomic imprinting. And so this suggests that this could potentially be something that might be involved in the dimorphic presentation that we see with MS, and would be certainly interesting to follow up on. So to kind of conclude what we've found so far, we've found that dense sequencing with the reference-free assembly method really allows us to identify high-resolution and potentially novel MS risk candidates that are not necessarily going to be on these genotyping chips. We find that stratifying by 1501, DRB 1501, can actually increase our signal clarity. And we see evidence of both shared and population-specific risk variation. And so, you may have noticed some of the effect sizes of these are not large, often like 1.2, 1.4, sorry, 1.1, 1.14, things like that. But given the strong evidence we see for potential biological impact, it seems like these are likely functional in MS. But unsurprisingly, we know with complex traits and complex diseases that we're not expecting to see the sort of very strong functional signal associated with Mendelian traits either. This also some of these loci are actually, there's quite a few pseudogenes, some antisense RNA, and some novel long non-coding RNA that are coming out of our loci as well, which again opens up another level of expression variation that we may not have been thinking about with MS previously. And then this sort of potentially long-range splicing regulation of class 2 is kind of another novel aspect that we're starting to think about. And overall, this really supports a potential for DRB1 1501 independent expression dysregulation in MS. And then moving forward will be PCR genotyping validation for these target SNPs that we've identified. We have some ongoing LD and haplotype analysis that we're still working on. And then we're hoping to also perform some ATAC-seq and maybe some expression testing for the target SNPs we've identified. I just want to thank everybody in the lab and Jill. A variety of people have been involved in this project and making it happen, from Danilo Augusto to the Oxenberg lab. We've also interacted quite a bit with the Norman lab and the Dilthey lab. I just want to say thank you so much to everyone, and I will take questions. Awesome. Thank you so much. I appreciate it. And thank you, Stephan, for a great talk as well. All right. Okay.
Thank you very much, Kristen. I don't see any questions in the Q&A box.
There's a question in the chat.
Is there one in the chat? Okay, let's have a look. Okay. There's Roger. Did you observe any other specific HLA allele associations in the DRB1-1501 independent MS cohort?
So I didn't get to mention it. So like I said, we did genotype the HLA genes separately, right, using NGS engine. And when we ran the association just on those second field genotypes, aside from the 1501 signal, when we accounted for that, we really didn't see anything else particularly significant in terms of the HLA gene associations, the allelic associations.
Okay, there's a question from Jim Kaufman. He says, thanks for the interesting talk, Stephan. Does your model work for genetic inversions as well? This is a question for Stephan.
Oh, from Stephan. Yeah, we have not considered that yet. But it would be something worth looking into for sure. And yeah, it would be, we've been talking about this as well, but once Stephan's method is done, it would be great to take that and apply it in this context as well to even improve our sensitivity further.
Yeah, I think Jim was wondering, I was asking that to Stephan, but I guess you probably answered it. Yeah, obviously working closely. So I didn't know about the African American work.
Mhm.
That's given this a big boost, presumably, because you've got an
Certainly.
independent kind of, everyone was banging away at European populations. And are there any other populations that provide some additional kind of variation, if you like, that you might look at? Or is it, presumably, if it's African American and European, it's worldwide in some, I mean, have you looked at Asian populations, for example?
Yeah, so the reason we worked with these two populations was because they represent the majority of the demographics that come to the MS Center at UCSF. And so we do have a few individuals from other populations as well, but it's so far hasn't been enough with this study to really be able to get statistical signal from. But yeah, that being said, it absolutely would be great to apply this to other populations. If we could take the target capture sequencing protocol, maybe get some more samples. The more we can apply it to, the more information we'll be able to get, certainly.
Okay, yeah, thanks. Jim Kaufman again says, how do you imagine splicing changing the specificity of the MHC presentation? Do these new SNPs you have identified also have an effect on other autoimmune conditions that is not antigen specific?
Yeah, that's a great question. I don't think we've gotten that far to looking at its potential impact on antigen presentation. Presumably there would be some if we're talking about differential splicing. I don't know off the top of my head what that would look like. As far as these SNPs specifically, I don't think that there is necessarily a ton of data on them yet anyways. Again, like part of the purpose is to expand beyond what's just on the genotyping chips, right? And so the majority of SNPs that have a plethora of functional data are likely going to be the more common ones that we see on those chips. So I haven't seen any yet for these in terms of splicing impacting antigen presentation function. But again, this is still early on, and we'll be doing probably a more thorough literature review for sure.
Okay, now Roger's got a question. You mentioned a higher odds ratio for MS in AA compared to EA, so an African American compared to the European population. Do you think other non-MHC genes contribute to MS susceptibility in African Americans?
Yeah, so I don't know that I would say that there's a higher odds ratio overall necessarily. Let me go back. So this is odds ratio down here on the x-axis. I think it's a relatively similar range of odds ratio overall that we're seeing between the two, between the European American and the African American. And sorry, the second half was non-MHC, non-HLA genes involved. Yeah, no, absolutely. So like for this example here, some of these are in regions associated likely with HLA gene expression, but then there are a variety of them as well that have nothing to do with HLA too. So it will definitely, I think, be a combination of both that we end up finding. And that's also seen by previous studies too, like the 2019 study found a handful of non-HLA associated regions as well.
Mm. Can I ask you one more question?
Sure.
Kristen, what's the EBV status? Has that been looked at in your populations?
That's an excellent question. Thank you for asking that. So, I don't think we actually have EBV data on these. This has been a study that started prior to when we really realized the role of EBV in MS. So I don't think we have actually captured that, but I suspect, as with most MS patients, the vast majority of them will be EBV positive.
Mm.
And the control's likely too, right? Because it's what, like 95% of the population has EBV in general. Interestingly, a couple of these, I think 3 or 4 of these loci actually also fall near regions that are differentially methylated by EBV itself. One of them, I think it was this one, I think it was the HLA-C one. This one actually falls in a site that EBV differentially methylates too. So I think there's definitely potential for some interaction with how EBV is also restructuring the chromatin architecture of these genes, or of the host cells.
Okay, that was a really interesting couple of talks. Very good session. So I think we're just about out of time, but I'd like to thank both speakers today, and Steve for setting this up. If you're still there, Steve, are you still there? Would you like to wrap up?
Oh, absolutely. Also want to thank Dr. Scholz and Wade for their presentations, and I want to thank everyone who attended this webinar. I think it went really well, and I certainly learned a lot of stuff that I find fascinating. I also want to add that the next SIP webinar will be held on April 29th. We'll have a single speaker. Dr. Marco Colonna will be presenting his full Rose Payne Award lecture from the 2024 ASHI meeting, titled Immunogenetics, Illuminating HLA-related receptors from alloreactivity to neurodegeneration. So I hope that you all will be able to attend that April webinar. And thanks very much for attending this one.
Thanks very much.